So, good morning students. In the last class, we were discussing about the structure of anther. Once again, I will explain because this is one of the important questions. Structure of anther. As I told you, anther, which is one of the bilobed structure, okay, which is a terminal part of stamen. Stamen, what it is means it is one of the male reproductive structure, okay, or which is a part of which is also known as the androecium. Here, this question will be asked for 5 marks. Explain the structure or draw a neat label diagram of a TS section of anther. See here. In transverse section, so in the structure of anther, we will study the TS, that is the transverse section of anther. Okay, sometimes they will ask only the diagram, sometimes they will ask explain the structure. If they are asking explain the structure, you have to explain each wall layer. Okay, so see here, first of all, this anther, which is also known as in the anther there will be presence of microsporangium. Microsporangium means that is the pollen sacs. Okay, what it is? That is the pollen sacs. See, whatever the anther, if you take a transverse section, it will be, the outline will be somewhat circular in shape. Okay, this is the transverse section of anther. Here, it is surrounded by 4 wall layers. How many wall layers? This is anther or the TS section of anther. It is surrounded by 4 wall layers. What are that 4 wall layers? Means the first is epidermis. Okay, second is endothelium. Third is middle layer. And the fourth is tapetum. Okay, fourth is the tapetum layer. Here, whatever the cells of epidermis, first of all, the epidermis, which is the outermost layer, okay, their cells are flattened cells and tangentially arranged. Okay, what is the function of this epidermis layer? Whatever the epithelial cells which are flattened in shape, okay, they are protective. They are protecting the particular anther, protective in nature. So this is about the epidermis. How the cells will be means it will be like this, flattened cells. Okay, this is the epidermis layer. Which is the outermost layer, which is made up of the epithelial cells, which is arranged, which are flattened cells and which are arranged tangentially. So you have to draw, you have to fill all these pollen sacs like this. And next, the second layer, just below the epidermis layer, there will be presence of a layer in which the cells are somewhat elongated and larger cells which are arranged radially. How they are arranged? It is arranged radially. See here, this endothelium layer. See, if they ask for explain the structure, means one point you have to explain one layer. In that, if you write this much, that is enough. Okay. See, in the endothelium, I will write here about the endothelium. Endothelium layer is sub-epidermal layer. That means which is lying just below the epidermis layer. It is one of the sub-epidermal layer of anther, except this, which is single layer, or in some of the plant varieties, it will be single layer or multi-layer. That means many layer in some of the plants, and in some of the anther it will be single layered. Here the cells are radially arranged. How the cells are arranged? The cells are radially arranged. Except this, there is a presence here the endothelium, the cells are radially arranged and the cells are elongated with a thickening, and that thickening is the fibrous thickening. The cells are radially arranged and they are somewhat enlarged form, that we are calling it as the elongated cells with some thickening will be there on the cell wall, that is the fibrous thickening. Okay, which thickening? That is the fibrous thickening. So this is the endothelium layer. Here I will draw the endothelium layer, somewhat elongated in this way. Larger cells will be there as compared to the epidermis layer. Here the cells are radially arranged in each microsporangia, that means the pollen sac. In some of the anther it will be one layer, and in some of the it will be varying. In all the anther, in all the crop plants, in all the plant species, it will be not similar. Okay. So what is this layer? This layer is the endothelium. Then next, the third wall layer, that is the, here I will write the third wall layer. Third one, middle layer. The middle layer, which is made up of 1 to 3 layers. How many layers? That is 1 to 3 layers. And the cells of these layers are epidermal. Cells of this layer are which cells? Epithelial cells. So we can write it as the cells of this layer are epidermal layer, and it will be, how many layers will be there of the middle layer? Means it will be 1 to 3 layers. Okay, the next here this layer, middle layer, it is going to degenerate when the anther is mature. When the anther is going to mature, at that time it is going to degenerate. Means degenerate at the time of, or during the, at the maturity of anther. When the anther gets matured, at that time its middle layers are going to degenerate. Maturity of anther. So here I will draw the middle layers. These are the middle layers. Okay, here I am drawing only one layer of middle layer or three. Then the innermost of these anther, this is the layer. The innermost layer of this, that is the tapetum. What it is? Tapetum layer. These are the innermost layer which is present in the pollen sacs. Okay, that is the tapetum layer. So here the tapetum layer, the cells of tapetum or the whatever the tapetum cells are there, they are elongated larger cells which has the dense cytoplasm, which will be having more than one nuclei, means 2 or 3 or many nuclei will be there. Okay, the next here, this is about the middle layer, epidermis and endothelium. Next we will see about the tapetum layer, which is the innermost layer. Fourth layer, tapetum. Here it is one of the innermost layer. It is having a larger cells with the dense cytoplasm, and with more than one nuclei. See, in one question, they will ask what is the function of tapetum layer? Function of tapetum layer. This question will be asked for 1 mark. Very important question for 1 mark for your theory. The tapetum layer, whatever the cells of tapetum layer are there, here I will write the function, cells of tapetum layer nourishes the developing pollen grain. This is the function of the tapetum layer. See here, whatever the tapetum layer are there, it nourishes means it provides the nourishment, nutrients to what? To the developing pollen grain. See here, first of all, the cells are elongated, somewhat larger cells with more than one nuclei, and it has a dense cytoplasm. So, inner side to this tapetum layer, there will pollen grains are going to develop. Okay. So, here there is a mass of cells inside this tapetum layer that it is known as the sporogenous tissues. First of all, this layer is the fourth layer, that is the tapetum. Inner to this layer, in the pollen sacs, there will be presence of a compact mass of cells or the tissues will be like this, where the cells are compactly arranged or closely packed, and this group of cells we can call it as the sporogenous tissues. What it is? Sporogenous tissues. Okay, see here. So, these sporogenous tissues only will give rise to the pollen grains. Okay, they are these cells, that is the cells of sporogenous tissues. It will give rise to the pollen grains. Okay, by taking the nourishment from the surrounding cells, that is the tapetum cells. So here you can fill this gap with the middle layer. This is the middle layer. Here there will be presence of sporogenous tissues. If, see in exam, they will ask if only draw the label diagram. If time is less, you can refer. One more diagram is there, which will be in a very short. As you can see at the center region of the pollen sac. See, these are the pollen sacs. What I had already told you in the last class, there will be presence of two lobes. In that each lobe, there will be presence of two pollen sacs or the microsporangia. At the center region of this microsporangia or the pollen sac, there will be presence of what? Compactly arranged cells, that is known as the sporogenous tissue. So, this sporogenous tissue will undergo into the mitotic division. See here. Here, I will write about the sporogenous tissues. Sporogenous tissues. Here, when anther will be young, when anther is young, at that time, the cells are compactly packed, compactly arranged. Okay, and occupies the center region of or center of pollen sacs. And the next thing, the cells of these sporogenous tissues, whatever the cells you can see there, that cells of sporogenous tissues will divide. The cells will undergo into the division. Which division? That is into the mitosis division. The cells of sporogenous tissue will undergo into the cell division, that is the mitosis. Okay. When they will divide mitotically, then whatever, for example, this is the cell. It will undergo into the mitosis division. This is the cell of sporogenous tissue. Then they will give rise to the pollen mother cell. Which cells are going to form? The daughter cells after mitosis. They will give rise to the pollen mother cells. Understood? Okay. See here. First of all, at the center region, there will be presence of compactly arranged cells. That cells, which is occupying the center of pollen sac, that cells will undergo into the mitosis division, or they will divide mitotically to which will give rise to the pollen mother cells. So further, next process after giving rise to the pollen mother cells, here a process is going to take place that is known as the microsporogenesis. After this, understood? Okay, till here, these are the 4 layers. The next, I had explained you about the sporogenous tissues. Okay, when the cells are dividing mitotically, they will give rise to the pollen mother cells. These pollen mother cells undergo into a process that is known as the microsporogenesis. So let's see about that one. See here, when the anther is younger, at that time the cells of whatever the sporogenous tissues will be there, the cells will be polygonal in shape. Okay, polygonal in shape, and they are compactly packed. When this anther is going to mature, okay, when an anther matures, at that time, see, when the anther is younger, at that time the cells will be the polygonal in shape and compactly packed. When the anther is going to mature, during this condition, what happens means the cells of sporogenous tissue, it will become rounded or round in shape. And next thing here, they will loosely arranged. There it is compactly arranged, here it will become loosely arranged. Okay, the next, what happens is, whatever, when the cells are going to loosely arranged, at that time the size of the pollen sacs are going to increase. What is going to happen when the cells are loosely arranged? Okay, it requires more space or the more area for loosely arranging. That is the reason the pollen sac size is going to increase. Okay, then the spore mother cells, whatever the pollen mother cells are here, for example, this is the sporogenous tissue in which the cells will become rounded in shape and they will become what? Loosely arranged. Okay, well, when the anther gets matured, so at that time, these cells, okay, these cells are known as the pollen mother cells. What it is known as? These cells are known as the pollen mother cells. So these pollen mother cells, for example, this is one pollen mother cells. This pollen mother cells, this will be diploid cells. Okay, this pollen mother cell, PMC, I will represent it with the PMC, that is a short form of pollen mother cell. This pollen mother cell will undergo into a division, that is meiosis. Meiosis means that is the reductional division, and it will form haploid cells. Okay, and these cells we can call it as the pollen grains. What it is? These cells are the pollen grains. Okay, see here, first of all, and one more thing. This meiosis, this will undergo into the two meiotic division. This is the first meiotic division. Here I will explain. This is pollen mother cell with a nucleus. Okay, this will undergo into the meiosis 1. and it will give rise to the two cells. Again, these two cells undergoes into the meiosis to second division, which will give rise to the four cells. And always remember, meiosis 1 division, it will be equational division. Sorry, meiosis 1, always it will be reductional division. And meiosis 2, always it will be equational division. Here, the pollen mother cell, always it will be diploid, which will give rise to the two haploid cells. During what? During meiosis 1 division. Okay. The next, these cells will undergo into the meiosis 2, which is similar to the mitosis. That means the equational division. So here it will give rise to the 4 haploid cells, and these 4 haploid cells are the pollen grains. What they are? These are the pollen grains. So see here, they will produce the 4 cells, which is known as the haploid spore tetrad. This is, or else we can say, because they are going to form the 4 cells. So what it is? This is known as the haploid pollen tetrad or spore tetrad. Better we can write pollen tetrad. Tetrad means that is the 4 cells. Okay. So after 2 divisions of the pollen mother cells, it is going to form the haploid 4 cells, that is the pollen tetrad. Next, here, what exactly the definition of microsporogenesis? I will write here. Microsporogenesis means it is the process of formation of microspores. Here, the microspores means that is the pollen grains. Okay, it is the process of formation of microspores, that is the pollen grains, from a pollen mother cell. From a pollen mother cells. Okay, through what? Through a division that is known as the meiosis. Through meiosis. And this process is known as the microsporogenesis. What it is known as? It is known as the microsporogenesis, where there is a formation of microspores, that is a pollen grain, from the pollen mother cell. As you can see here, this is the pollen mother cell, from which a pollen grain or the spores are going to form, which is known as the tetrad, pollen tetrad. Later on, we can call it as the pollen grains. So, when the anther matures, the middle layer and the tapetum layer are going to degenerate. Okay, in the TS section of anther, you had seen there will be 4 wall layers. In that, the second layer, the third layer is the middle layer, and the innermost layer is the tapetum layer. So, when the anther gets matured, at that time, the 2 layers are going to diminish. Here I will mention, when anther matures, tapetum and middle layers get degenerate because they have to dehiscize the anther. Dehiscize means they have to break the anther. Okay, so the products of the tapetum layer and the middle layer, it is going to be utilized by the developing pollen grains. Whatever the cytoplasm, whatever the nutrients which are present in the middle layer and the tapetum layer, that is going to utilize by whom? By the developing pollen grains. Okay, so why these two layers are going to degenerate means, see here, if this is the anther, when the anther gets matured, the wall of the anther has to break down, or it has to dehiscize. So for this dehiscence of wall of anther, what happens? The tapetum layer and the middle layer, it is going to degenerate. If it is going to degenerate, then only the mature anther is going to break it down. Then it will release a yellow colored powdery mass. Yellow colored, a powdery structures or the spores, which is known as the pollen grains. What it is known as? It is known as the pollen grains. So here, as I told you, there will be formation of pollen tetrad. What even by this pollen tetrad means? Let's see here. First of all, how the pollen mother cell will be there means, this is the pollen mother cell, which will be the diploid. Okay, this pollen mother cell, first it will undergo into the meiosis 1, which will form the 2 cells. Okay, then it will undergo into the second division. Here I had written meiosis 2, then it will form 4 cells, and the structure we can call it as the pollen tetrad. Okay, this pollen tetrad, which will further give rise to the pollen grains. This pollen tetrad, when the anther gets matured, at that time this layer is going to disrupt, which will give rise to the 4 pollen grains. Each pollen mother cell will give rise to the 4 pollen grains. So next we will see about the pollen grains. Next topic, that is the pollen grains. See, the diagram around the pollen grain, this question will be asked for 2 marks. Explain the structure of the pollen grain. Here, next topic, that is the pollen grain. Here, what is pollen grain means? It is a yellowish powdery, means it will be powdery structures released by the matured anthers, or released by the splitting anthers. Whatever the anthers will be there, when it gets matured, that walls are going to disrupt and they will release some yellow. If you touch any flower, at that time on your fingers there will be yellow color powder going to stick. What is that? Means that is the pollen grain. If you take that powder and observe under the microscope, you can see the different shapes and structures of that particular pollen grains of different plants. So what is this pollen? First of all, it is a microscopic structure. Okay, you cannot see with your naked eyes. It will be so much small and it has a fascinating shapes and structures. So here the pollen grain, it is haploid. What it is? It is haploid. But the pollen mother cell is the diploid. Okay, the pollen mother cell are going to give rise to the pollen cells. That's why that is diploid and this is the haploid. And here the pollen grain represents the first stage of male endophyte. It represents the first stage of male gametophyte or the male gamete, what we call. So it is going to represent that first stage. The next here the pollen grain, how much will be the size means the size of the pollen grain is it measures about 25 to 30, so 25 to 50 micron in diameter. Okay, how much is the size of pollen grain? 25 to 50 micron in diameter, and it has wall layer. The pollen grain has wall layers. How many wall layers are there? That is 2 wall layers are there. Okay, so let's see about that wall layers. First of all, this is the outer layer or outer wall layer, outermost wall layer. This outermost wall layer is known as the enzyme. Enzyme, which is the outermost or outer layer of pollen grain, and its surface is rough surface. Here, the enzyme surface is having a very rough surface due to the presence of some ridges or outgrowths. Okay, you can see somewhat outgrowths or ridges on this layer, that is known as the enzyme layer. And this enzyme, it is made up of, here I write, enzyme, which is the outermost layer, which is having a rough surface or the ridges. Okay, so it is made up of a biochemical, and that biochemical name is known as the sporopollenin. What it is made up of? It is made up of a chemical substance that is known as the sporopollenin. Next, inner to this layer, and one more thing here, at some region there will be a thin formation of the enzyme layer, and this layer, which I am representing it with the imaginary line, this region is known as the germ pore. Okay, what it is known as? Germ pore, from where the pollen tube is going to emerge out. Okay, about pollen tube you will come to know in the further topics. Then here, inner to this layer, inner to enzyme layer, there will be presence of a layer, wall layer, which is known as the enzyme. What it is known as? It is known as the enzyme layer. Okay, so this enzyme layer, it is one of the protective layer of the pollen grain, and enzyme layer, enzyme layer is there. It is, if you talk about the chemical nature, it is pectocellulosic. There will be presence of a plates of enzymes, enzymatic proteins. This enzyme layer, it It has a plates of enzymatic proteins. Some proteins will be there which is present in the form of what? Plates. Okay. So this is about the two layers. In the next class, I will explain about the cells which are present in the pollen grains and some more things about the structure of pollen grains. Okay, thank you.